In this video, we're going to learn about adaptations that apply to many of the organisms that we're studying in the diversity unit. So in that sense, this video is a bit of an evolution connect between our diversity unit and the evolution unit. So we'll take a look at adaptations that apply to halophilic archaea and some other organisms that we studied in the diversity unit. So just a key point to remember is that natural selection is the only mechanism of evolution that consistently leads to adaptive evolution. It results in populations that are better fit for their environment. So we're going to take a look at some of those key adaptations that exist today in many of these organisms, especially in some of the unicellular organisms that we often don't spend as much time studying in the grade 11 biology unit. So before we go into some of the adaptations, we need to recall a process that you learned about maybe in grade 10, and if you didn't, you'll learn about it here. This process is called osmosis. Osmosis is the diffusion of water across a semi-permeable membrane. Diffusion just means a movement across a membrane. So, for example, your cells have a membrane, and when something moves across that membrane through pores, for example, I should have drawn pores here, through these little pores here, that's diffusion. Osmosis specifically is the diffusion of water across a semi-permeable membrane. Now, semi-permeable means that the membrane lets things through, but not all things through. So permeable means to let things through, but it's semi-permeable. So when water goes through a semi-permeable membrane, that is an example of osmosis. So here we have a U-tube. It's a tube shaped like a U. And inside you have some water and also some of these green dots here that represent solutes. Now solutes can be sugar, it can be salt, something dissolved in the water. Typically, water is going to move from an area where there's low solute to an area where there is high solute. So water moves from low solute to high solute areas. And the reason that is because solutes tie water up. Water is attracted to many solutes. So on this side over here, where you have a lot of solutes, water is tied up. It's not free water anymore. So there's fewer free water molecules when there's more solutes. So the idea is that water will flow from an area where there is a lot of free water, let's just say a lot of free water, to an area where there is little free water. And that coincides with, a lot of free water coincides with low solute, and little free water coincides with high solute. So low solute has a lot of free water, so that free water will flow to the side with high solute, which has low free water. Typically, we represent the solutes amounts using concentration, and we use the letter M, molarity, for concentration. A higher M means a higher concentration of solute in that solution. So if I have 2M versus 1M, 2M is more concentrated in terms of solutes than 1M. So 2M would have lower free water compared to 1M. 1M would have more free water. So that's how osmosis works. Osmosis is just a movement of water from one side of a membrane to the other. Water will flow from the area where there's less solute to the area where there's more solute, because where there's less solute, there's more free water, and where there's more solute, there's less free water. And solute can be sugar, salt. And what I want you to notice is that it's a semi-permeable membrane here. Only the water is going through, but the solute particles are too large, so they aren't going through. So many times in cells, water will go through, but other particles won't be able to make it through. So I've mentioned this before, when there's solutes present, there's less free water because the water is busy dissolving the solute. So here, this water is not free anymore. Over here, there's fewer solutes, so there's more free water. It's not busy surrounding the solutes. So in this case, water would flow from the right side to the left side across the semi-permeable membrane. And it keeps flowing until an equilibrium is reached. It keeps flowing until an equilibrium is reached. So um, the idea is that the water will flow until the concentrations are equal on both sides. Now, what I want you to understand is that when the concentrations are equal on both sides, the water actually does not stop. So it doesn't completely stop. It's just that the flow will be as if there's no net movement. So for example, if concentrations are equal, when one water molecule goes to the right, another one will go to the left. And so overall there'll be no net movement at equilibrium. There's still movement. Particles don't just say, all right, stop guys, we're done moving. No, it's just there's no net movement once that equilibrium is reached. So let's try to answer some of these questions. So remember, bigger number means more concentrated in terms of solute. So we have a bag over here. It's a semi-permeable bag. It represents a cell model. And then we have a solution over here. The solution has 0.6 molar. The bag, the solution on the inside of the cell, is 0.2 molar. So the higher solute concentration is the solution over here, higher solute concentration. This is a lower solute concentration. Lower solute concentration, higher solute concentration. So this bag is called a dialysis bag. We have to determine where the water will go when osmosis happens. Will it go from bag to solution on the outside, this way, A, or will it go the other way, this way over here? So the question you have to ask yourself is, where is there more free water? There's more free water when there's lower solutes. So there's more free water here. Because there's more free water here, the water will flow from the bag out to the solution in the beaker until the concentrations will become closer to each other. So as you get water leaving the bag, the water that leaves the bag will diminish the concentration of the beaker on the outside, and an equilibrium concentration will be reached, and at that point water will just flow in and out at the same rate. So at this point we're going to say the dialysis bag, the beaker, which one has a higher concentration? Well, we know it's the beaker that has a higher concentration. Now, still with our same question here, if the water flows out via osmosis, what should happen to the size of the bag? Well, the bag will get smaller because water has left it. So you should see your bag start to shrivel up a little bit. And the reason that the water left it is because, again, the dialysis bag had more free water than the beaker. As a result, the water flows from the bag to the beaker. So you should be able to predict which way water will flow based on the concentration of the solute that you see in the problem that's provided. So you can try this one over here. You can predict which way solutes will flow. So let's take a look. Higher concentration of solutes in the bag, lower concentration of solute in the surrounding solution of the beaker. That means the bag has less free water than the beaker. So water will flow from the beaker to the bag, and that will dilute, meaning lower the concentration of the solute as water is added in. Dilute means basically to water down. And then once equilibrium is reached, once they have the same concentration, then the water will keep moving, but just no net movement, meaning water will enter and exit at the same rate. If one goes out, one comes in. That's only once equilibrium is reached. So the dialysis bag, remember, is semi-permeable, meaning water can go through but solutes won't. So we know that the dialysis bag has a higher concentration, and that's what I just explained. So because the dialysis bag has a higher concentration of solute, we know that the beaker is actually going to have more free water this time. As a result, the water will flow from the beaker to the bag, and the bag, as opposed to shrivel up, will get bigger this time. So you'll see this happening, and the bag will get bigger because more water is flowing into it. So over here in this case, which one has a higher solute concentration? Well, if you look at this carefully, they both have the same concentration. So, and we still have a dialysis bag that's semi-permeable, so only water can go through it. So the dialysis bag or the beaker has a higher concentration? Well, none. They both have the same concentration this time. And so what will happen to the size of the bag? Well, let's take a look. The dialysis bag and beaker, well, they both have equal amounts of free water, let's just say. And because of that, well, it's actually both of these that will happen at the same rate. So as one water molecule exits, another one will enter. It's one for one, one for one. So even though there's water moving, it's as if nothing's leaving or nothing's entering. So the bag won't get bigger or smaller. It should stay the same size because they are already at equilibrium. They are already at equilibrium. So you should go back and practice analyzing these situations to see if you could predict which way water would move. We're going to make this connection to evolution in just a minute. So now that you know osmosis, we're going to take a look at different types of solutions that cells can be found in. Many cells live in aquatic environments, and environments where there's watery solutions, and those solutions, because they're solutions, they have solutes in them. So there's three main types of solutions that you can see that a cell can be found in. So we're going to take a look at what that looks like first for cells without a cell wall, and then we'll take a look at what this looks like for cells with a cell wall. So there's hypertonic, hypotonic, and isotonic solutions. Here we have a cell, an animal cell, a red blood cell, that was put into an isotonic solution. Isotonic means that the concentration of solute on the inside of the cell is the same on the outside. So for example, we have 2M on the outside and 2M on the inside. So water enters and exits at the same rate. For an animal cell, this is a great condition to be in because your cell doesn't get too big and your cell doesn't get too small. So isotonic is great for cells where you don't have a cell wall. Now there's two other types of solutions a cell can be in. You can have a hypertonic solution or a hypotonic solution. So let's look at the word hypertonic. Hyper means above. So a hypertonic solution has a higher concentration of solutes than the cell that enters it. So for example, here we'd have hypertonic, 2M, and the cell might be 1M. So because there's a higher concentration of solute on the outside of the cell, water will leave the cell. And for a cell without a cell wall, this is not good. It's going to start to shrivel up. And so the cell becomes shriveled up in a hypertonic solution. So if we put, let's say, we hook it up to a machine in the hospital, to an IV, and we put a solution that's hypertonic to your cells, we run the risk of shriveling up your cells because water will leave your cells. It'll basically, think of it this way, there's a lot of solutes here. And when there's a lot of solutes on the outside, I like to use the saying solutes or salt sucks. Solute sucks. It sucks the water out of you. It sucks the water. The other type of solution is called hypotonic. It's a solution where the concentration of solutes is lower than the concentration of solutes inside of the cell. So in this case, it's the other direction. Because you have a lower concentration of solute here, you have more free water. And so what happens is water will flow into the cell. And this might seem good at first, but if too much water flows into the cell, it gets bigger and bigger and bigger, and then it bursts. And that bursting is called lysed or lysing. Okay? So for an animal cell or a cell in general without a cell wall, so animal cells, some protists that don't have cell walls, the isotonic condition is the best. The hypotonic leads to lysing, and the hypertonic leads to getting shriveled up, which is not good. So think about when we put salt on our roads during the winter to melt ice, think about what that could do to some plants and their health. Think about if we're creating hypertonic solutions or hypotonic solutions. Think about it for a minute, pause the video, and come up with an answer. And hopefully you came up with the idea that, yes, we're creating hypertonic solutions for the plant's environment, and that's sucking water out of the plant. I know a plant has a cell wall, but for plants and organisms with cell walls, hypertonic solutions do a similar effect where they cause a cell to get kind of shriveled up, and we'll take a look at that in a few minutes. So here we see a summary of the different conditions with a view of the solutes in more detail. So you should know how to draw and represent the different types of solutions for and how they affect cells without cell walls. So here we have an example of a protozoan, so an animal-like protist, specifically called the paramecium or paramecia plural. It's a ciliate. It has cilia to move around with. And so it has some interesting adaptations that allow it to survive in some interesting environments. So because this doesn't have a rigid cell wall, it is really, really vulnerable to changes in its water, to changes in the water concentration within it, or I should say, in the amount of free water within it. So the paramecium actually has two adaptations that allow it to withstand changes to the water that comes into it. The first is that its plasma membrane is actually less permeable to water. So if it's ever in a hypotonic solution, yeah, water will enter it, but not as quickly as other cells that have a higher permeability to water. So water will enter it, but not as quickly, so it won't run the risk of bursting. And the reason that it has this adaptation is that these are found in a lot of freshwater environments where there's low solute concentration compared to the cell on the inside. And now another adaptation it has is the contractile vacuole, which is something you would have seen in the protist lesson. When too much water does come in, this vacuole takes in that water and then pumps the water out. So that way the cell doesn't run the risk of bursting. So these two adaptations is what makes this paramecium really perfect for its freshwater environments, because it's living in an environment that's hypotonic to the cell itself. And so the adaptations allow for what's called osmoregulation, which is basically the control of water balance within the cell. Now the reason that those adaptations were required for the paramecium is because it does not have a cell wall. When you have a cell wall, the different solutions affect you in different ways. For animal cells and cells without cell walls, isotonic was the best condition to have. But for cells with cell walls, like plants, prokaryotes, fungi, and some protists, isotonic is not necessarily the best condition. So let's take a look at why that is. So again, we have the three conditions here: hypertonic, hypotonic, isotonic. I think I checked those off incorrectly. Iso, hypo, and hyper. And so let's go take a look at an isotonic environment. So this is an isotonic environment here. 2m and 2m. Water enters and equals at the same rate. For plants and organisms without cell walls, this is not the greatest because, or sorry, with cell walls, this is not the greatest condition because the plant, for example, needs to be firm. So if you have a plant with a stem, if it's not firm, it'll fall over easily and be very flimsy, and that's not good for the plant structure and integrity. So in an isotonic environment, your plant is actually flaccid. It kind of just plops over, and it looks frail and weak. When you take the plant, however, and you put it into a hypotonic environment where you have a lower concentration on the outside compared to the inside, water flows in. And with animals and other cells without a cell wall, the plant could burst. But because there's a cell wall in this case, so let's pretend this is a plant here, and let's not pretend it is a plant. With the cell wall in this case, what happens is the water won't burst the cell. The cell wall will push back inwards and create a firm cell, and so that leads to a turgid environment. It's very firm, it's a better solution, it's a better environment for the plant, and it can now stand on its own and have more structure and integrity. So for organisms with cell walls, especially plant cells, the hypotonic environment is the best. Isotonic is you need a better environment, and the hypertonic, that's still bad. So hypertonic, what happens is so let's have over here our 3m versus, 2m over here on the inside. The water leaves and the cell kind of shrivels up, but its membrane stays attached to the cell wall here, and so eventually it will cave in inwards if it shrivels up enough, but it's really the insides that are shriveling up. And we call this, for cells with cell walls, we call that plasmolysis, or it's plasmolized. In the case of the animal cell, we simply said shrivels up, but in the case of the plant cell, we say it's plasmolized. So turgid is the best environment. If you become isotonic, now you're kind of flaccid, and the next step is if you become in a hypertonic environment, you become plasmolized. You would shrivel up all the way, but those attachment sites at the cell wall prevent the cell from completely collapsing inwards, at least initially. So here we can see the situations of the different solutions. You can see that this is the best environment for the plant. It's now turgid because that pressure of pushing back by the cell wall makes it nice and firm. Isotonic, not enough pressure pushing back, so it's flaccid. And then over here, plasmolized, it just kind of shrivels up on the inside. So here we can see an example of a nice turgid plant placed in a turgid environment. It's nice and firm. You can see that nice coloration. Here we can see a plant that was placed into a sugary solution. So there was a sugary solution, like corn syrup. The plant was placed in there, and because the solution is just sugary and has a high molarity, 3M, and this might have had a low molarity, like 1M, a whole bunch of water started to leave it. And so it became plasmolized. In fact, you can even see that in the plant cells here. Here they take up all the space, the membranes and everything is nice and colored in for the cell. Whereas over here, if you look at this environment that's hypertonic, you can see the membrane retracting away from the cell wall because it's starting to undergo plasmolysis. So it's in a hypertonic environment. So when you put that salt out in the winter, you're creating that hypertonic environment for the plant cell. So there's some interesting applications relating to this. If we look at the term salt curing is when you basically cover your food in salt so you can kill bacteria. And bacteria are organisms with some pretty tough cell walls. So what happens with the salt curing, and maybe you can try to explain it yourself first. Pause the video, come back to it after, see if you had the same explanation. So salt curing creates a hypertonic environment on the outside. Hypertonic environment on the outside, so it might be, for example, 3M. And on the inside, for the bacterial cell, you might have 1M. And so it's going to suck a bunch of the water out of the bacterial cell, and then a bacterial cell will eventually become, let's say, plasmolized and shrivel up, and it could eventually die if you put too much salt. So that's called salt curing your food so that you can kill some harmful bacteria with some of those tough cell walls. So this is the idea of how salt curing works. So you can take a look at the hypertonic environment. Now you should be able to propose some examples of adaptations that might allow a bacterial cell to survive in these environments. So maybe there could be a mutation that leads to some kind of membrane that's more permeable to water. Maybe there's a mutation that leads to some kind of protein that lets water leave when there's too much in, that lets water, I should have said the other way around. See, so already I made a mistake there. So maybe you can make a protein that lets water come back in once there's not enough water in the bacterial cell. So think of adaptations that will allow the bacterial cell to survive in a salt cure environment. Kind of like your natural selection assignment that you were doing. Here are the different conditions of the you could see the animal and plant cells in, so you can compare them side by side, know which condition is better for the animal cell, know which condition is better for the plant cell and why that is, and then know the dangers of being in these different conditions, but more importantly, know the adaptations that allow some organisms to live in these hypotonic environments or maybe hypertonic environments. Think about the extremophiles that are able to survive in those environments. Speaking of extremophiles, let's take a look at a halophile. A halophile is an extremophile that actually likes salty environments. It has adaptations that allow it to survive in the hypertonic environment. So this halophile is a prokaryote, so we know it has a cell wall. It's an archaea specifically. It's an extremophile. It lives in these very extreme salty conditions, so maybe very salty oceans, that would normally dehydrate some of the they normally dehydrate other cells. So normally if you're in these salty conditions, water will leave you like crazy and then you'll shrivel up or you'll plasmolize. But these halophiles have some biochemical adaptations that allow it to survive in the environment. So it has these things called active salt transporters. They're basically proteins in the membrane that will pump potassium ions into the organism. So let me show you what I mean by that. So we have a very salty environment, and let's say the salty stuff comes from potassium ions out here. Normally, water would want to be sucked out because it's very salty, but this cell has a pump that puts some of the K plus into itself, so it lowers the difference in concentration between the two sides, so that way less water is leaving the cell. And so that's one of the biochemical adaptations that this halophile has to surviving in a hypertonic environment. So we saw adaptations of the paramecium to a hypotonic environment. We saw adaptations of the prokaryote, the halophile, to a hypertonic environment. So again, we can see natural selection resulted in organisms that were good matches for their environment. If the organism did not have this pump, it would just not survive in this environment, so it's not there. If the organism does have it, then it can survive, and it is there. And this was a result of a random mutation that allowed that protein to be embedded within the membrane. So over here you can see some examples of halophiles in some salty environments, and they're able to survive in those salty environments. Whereas if you have a non-halophile organism in a salty environment, the water will come out and you'll have plasmolysis if it's a cell-walled organism, or shriveling up if it's a non-cell-walled organism. So you should complete the learning goals for this PowerPoint, and then you should go in and complete a gizmos. There's a gizmos that corresponds to the adaptations of some organisms in different types of solutions. Complete the gizmos, the package, and complete the quiz with that gizmos, as I will be checking to see for its completion, and I will be checking the package as well.